Dear students, Module 29, Innate and Adaptive Immunity and Immune Response. The main objectives are to know about the concept of innate and acquired immunity, to understand the barriers and the proteins of innate immunity, to explain the inflammation response, to know about the humoral and cell mediated immunity, to know about the immune response of the cells of the immune system. Introduction. Many dangerous microorganisms will attack our body like bacteria and viruses. They enter our body and damage the tissue. Our immune system plays a significant role in protecting our body against those pathogens. Innate immunity is less specific and uniform in all members of the species. Innate, as the name suggests, it is by birth. The disease resistance mechanism is already present in our body by birth. It is the first line of defense during the early stages of the development. Innate immunity can be shown by phagocytic cells, namely macrophages and neutrophils. They engulf the foreign bodies to kill them, called phagocytosis. Example, skin, which is one of the major barriers for the pathogens to enter into our body, and some of the antimicrobial components helps in innate immunity. Adaptive immunity is the more specific type of immunity. This immunity comes into action only if there is any antigenic activity, that is, the pathogenic attack. When a foreign body enters the cells specific to that antigen, the immune response activates and tries to kill that antigen. At the same time, there will be a development of memory cells where if the same pathogen attacks for the second time, the action will be very quick against those antigens. Hence, in this episode, the following aspects are going to learn. A. Barriers against the antigens in innate immunity. B. Inflammatory response. C. Proteins of innate immunity. D. Humoral and cell mediated immunity. E. Immune response. A. Innate immunity, barriers against the antigen. There are several pathogens or foreign molecules which invade into the body through any means. It can enter while breathing and eating. These show non-specific innate immunity. There are mainly 4 types of barriers, namely anatomic, physiologic, phagocytic and inflammatory barriers. Anatomic barriers. There will be some structures and fluid in our body which prevent those pathogens from entering into the body. Skin is one of the critical obstacles to pathogens. It covers our entire body and prevents the easy entry of pathogens. Mucous membranes are the second important barrier after the skin. Normally, mucous layer present as a lining to the respiratory tract and mouth. Ear wax and cilia are present inside the respiratory tract, act as barriers. When any dust or small organisms enter through the ear or through the nose, the wax which traps those particles inside the ear and cilia, which helps in pushing those particles of the respiratory tract. Skin, mucous membrane, ear wax and cilia gives the first line of defense. Physiologic barriers. There are few physiologic barriers like low pH, which create the acidic environment in the stomach and kills most of the bacteria. Body temperature is favorable for survival of many bacteria and viruses. Soluble molecules such as lysozymes, complement proteins and interferons play an essential role as the physiologic barrier. Phagocytic barriers. This type includes the phagocytes which will engulf the pathogens and digest the pathogen particles. Phagocytosis is a type of endocytosis where the phagocytic cells will extend their membrane around the pathogen and engulf inside, where the pathogen particles will settle inside the vesicle to form the phagosome. The phagosomes will then fuse with the lysosomes. Lysosomes are the membrane bound organelles, consist of about 40 different hydrolytic enzymes inside them. When phagosomes fuse with the lysosomes, the enzymes will start digesting the pathogen particles and the digested particles are released out of the cell. Inflammatory barriers. This will be shown up by the cells of the immune system. There we see a cascade of changes that occur in the injured area. The inflammatory response is by the immune system against the pathogen. B. Inflammatory response. Inflammation is the immune response shown by our body where the cells of the immune system gather together in the place of injury and show the immune action. It is the central part of the innate immune system. The infection which can lead to the proliferation of the pathogens entered inside our body. The wound in any part of the body shows inflammation in a while is the natural process by the immune response is called the inflammatory response. Naturally, the inflammatory response shows the sign of redness, swelling, heat and pain in the region of injury. As the antigen enters the body, the damaged surrounding tissues start producing chemotactic factors. These chemotactic factors are responsible for the vasodilations, that is, the blood capillaries begin to enlarge near the injured area and the movement of the blood will be easier and faster near the damaged tissue. This will result in the reddening of the injured area and the increase in the heat of the affected area. When the blood capillaries increase their size in diameter, the fluids and other important immune cells get released into the affected tissue areas, and this will result in the swelling of the tissues. The process of release of immune cells from the blood capillaries is called extravasation. For example, the monocytes, they migrate into the bloodstream by attaching to the wall of capillaries and enter inside the tissue through the capillary membrane, and in the tissue they act as the macrophages. These macrophages go near the wounded region, engulf those antigens, and inside them form the vesicle with the antigen. These are termed as phagosomes. As mentioned above, the chemotactic factors are the chemicals which are produced by some of the cells of the immune system during the injuries or any other processes. These play an essential part in the initiation of the inflammatory response. As the name suggests, these chemical components attract the cells of the immune system near the regions of injury. The common and more importantly studied chemotactic factor is histamine. It plays a role during tissue injury. They can act as the ligands which go and bind to the receptor on the membrane of the capillaries and responsible for the vasodilations. There are many other chemotactic factors like C-reactive proteins, peptides like kinins, which are usually present in blood as an inactive form and get activated by any responses. C. Proteins of the innate immunity. Along with inflammation, there are proteins which play an essential role in the innate immunity. These includes cytokines, complement system and collectins. 1. Cytokines. Cytokines plays an important role in both innate and adaptive immunity. In the case of the innate immune response, some of the cytokines called interferons help in the activation of the cells of the immune system to fight against the pathogens. As the name suggests, they mainly interfere in the replication of viral particles. Other cytokines called interleukins are also triggered the body temperature or play a role in causing the fever, so that the temperature can kill some of the bacteria and viruses. The higher body temperature can also reduce the microbial growth inside the body. There are many groups under interleukins based on different structural characteristics. Tumor necrosis factor are also one of the type of cytokines that helps in the destruction of bacterial toxins. 2. Complement system. They consist of proteins which increase or complement the action of the immune system. Mainly it includes plasma protein, where the ability of the antibodies against the pathogens get increases. Some proteins help in the destruction of the plasma membrane of bacteria. 3. Collectins. They also protect the body from viruses, bacteria and yeast. Collectins mainly differentiate these pathogens by the surface molecule present on those pathogens which are different from the human cells. D. Humoral and cell mediated immunity. When an antigen gets engulfed by the macrophages, those cells act as antigen presenting cells, which are also called APCs. The antigen can be a pathogen or part of the pathogen like flagella of the bacteria, part of the virus, etc. Those part of the pathogens are called as the free antigens. These APCs will showcase those antigens to the rest of the cells of the immune system. Mainly, the APCs will activate the helper T cells. The activated Th cells can activate either B cells or the cytotoxic T cells. Interleukins 12 and interferons are the cytokines released by these cells of the immune system helps in the activation process. The free antigens can directly activate the B cells, then those B cells get converted into the plasma cells. Plasma cells will start producing the antibodies specific to those antigen. They can also direct to kill the antigens by any other complement system or phagocytic cells. At the same time, if the antigen is attacking for the first time, then along with plasma cells, the memory cells are also formed for the secondary immune response. This shows the humoral type of immune response. The macrophage will secrete many types of chemical molecules, that is cytokines. Interleukins 12 is one such cytokines act on Th cells. Interleukins 2 secreted by the T cells and acts on T cell itself. Another cytokine called interferon gamma to get released by the T cells. There will be the proliferation of more T cells by the action of interleukin 2 and also other natural killer cells get activated. IL-4 and IL-5 get released from the other cells by the action of interferon gamma, and those interleukins act on the B cells. B cells, as mentioned above, start the humoral immunity. IL-1 produced by the T cells helps in the activation of new T cells. These new T cells get proliferated to form more Ts cells. The Ts cells will activate the cytotoxic T cells. When they get activated, cytotoxic T cells start to kill the affected target cells. The cytotoxic T cell can also get activated directly by the signals produced from the affected cells. When the antigen enter into the cell, they make specific changes in the membrane of the cells in the body. Those changes in the membrane will act as a signal to the cytotoxic T cells. Activated cytotoxic T cells can then kill the infected cells. This shows the cell-mediated type of immune response. E. Immune response. The immune response is the main role played by our body system for any external or internal immunogenic attack. Immune response varies at any age point. During the infant stage, the development of the immune system takes place. At this age, body cannot fight against all types of antigens. There will be some maternal antibodies transferred to the infants can give protection. After the 6 to 12 months of age, the immune system gets active. When the individual gets older, the capacity of the immune response will get reduced. The immune response includes the following two types: innate and the acquired immune response. The innate immune response comes into picture by birth. As mentioned earlier, this type of immune response has no specificity against the antigens. They will show the vast and quick responses to the antigen attack. There will not be the formation of any memory system by this response. Acquired immune response work more specifically. The response will be lasting for more time. It will just increase the immune response of the innate type. There will also be the production of memory system, helps the cells to acquire the power of acting very quickly in the next encounter of the same antigen. The immune system should be ready to face the intrinsic actions of the body. When there are some mistakes in the cell machinery, can lead to some mutations. Immune responses also have the power to suppress the tumorigenesis. They can also show the interactions of the immune system with the cancer cells. There can be epigenetic changes occur, result in the alteration in the expression of the gene without the differences in the DNA sequence. The cell-mediated immune response is mainly regulated by the gene called immune response gene. Later, it came to know that the immune response genes are nothing but the MSC genes. MSC genes codes for the cluster of proteins which show up as the critical surface protein molecules. Earlier, they were termed as the His2 antigen or histocompatibility to antigens. Later, in mouse, they reported as the MSC molecule. When the viral particle infects the cells, the MSC class I receptor will show some particles of the virus to the immune system cells. MSC class I will mainly detect by the CD8 type of T cells called cytotoxic T cells or killer T cells. The role of cytotoxic T cell is to kill the target cells. The target cell receptors will interact with this antigen bound with MSC1. There will be some cytoskeleton rearrangements in T cells which make the membrane bound vesicle inside the T cells to fuse with the T cell membrane. Importantly, the release of chemical factors takes place like porphyrins and granzymes from the vesicle. Porphyrins will form the pores in the target cell membrane, and granzymes will enter inside the target cell where it degrades the cell components and activates the apoptotic pathways. MSC molecules are divided into mainly three types. They are MSC class 1, MHC class 2, MSC class 3 molecules. MSC class 1 and MSC class 2 play their essential role in the immune response. Usually, these MSC molecules bind to the antigen by the extracellular NH2 domain. Then the antigen are exposed out to the immune cells. MSC class 1. All the nucleated cells have the surface bind molecules as MSC class 1 units. These molecules consist of two polypeptide chains, namely alpha and beta 2. Alpha chain is the transmembrane polypeptide, consists of alpha 1, alpha 2 and alpha 3 as the three domains. Beta 2 is an extracellular myoglobin protein molecule. It attaches to the three domains of the alpha chain non-covalently. Beta 2 microglobin is not encoded inside the MSC molecule. Alpha 3 and the beta 2 are immunoglobulin like molecules. The working of these MSC class 1 molecules helps in the cell mediated immunity. These molecules will act as the surface bound epitopes on the nucleated cells. The CD types of T cells or cytotoxic T cells will recognize the class 1 MSC molecule. The T cell have the CD8 receptor along with the T cell receptor. These molecules consist of two polypeptide chains, namely alpha and beta. Alpha chain possesses alpha 1 and alpha 2 as the two functional domains. Same way, beta chain consists of B1 and B2 as a functional domain. MSC class 2 type of molecules are recognized by the CD4 type of T cells or the helper T cells. Here, both the chains are encoded within the MSC molecules and are associated with each other non-covalently. Alpha 2 and the beta 2 are the Ig-like molecules. Alpha 1 and beta 1 are the outermost domains present. The antigen presenting cells will present the antigen bound to the MSC class 2 receptor molecules on the membrane. The CD4 receptor molecules on the helper T cells will recognize those cells and bind with the antigen bound to the MSC class 2 molecules. During the binding, the new helper T cell will form the memory helper T cell. For the secondary response or the effector helper T cells MSC class 3, this molecule will not show its role as the class 1 and class 2 molecules in the immune response. It is different from other molecules and includes the secreted proteins. Many secreted proteins act as an activator of the cell or the immune pathway. The proteins like cytokines and the proteins of the complement system have their role in the immune function. To conclude, innate as well as adaptive immunity play an important role in immune response, and both types have differences and show quick and long lasting immune responses. Thank you.